This is a brief review of the coagulation cascade. It's not going into all of the fine details of what happens throughout the coagulation cascade, but just a reminder of what happens, why it's there, and how things could go wrong. So let's review a few key things about the coagulation cascade. Now, we have to have a really clear understanding of the fundamentals of this cascade in order to understand DIC. So starting here, why does this coagulation cascade exist? What is it again? Well, the coagulation cascade is kind of like a chain reaction that happens in the body, and actually, as you can see from this image, a pretty complicated chain reaction. And at the very end of that chain reaction, fibrin is formed. And wait, what's fibrin again? Well, fibrin is this small protein substance, and these fibrin substances, as they're produced by this coagulation cascade chain reaction, they start to connect to other fibrin substances and become fibrin strands, and these strands begin to cross-link and actually hold a clot together. So think of it this way. Once the coagulation cascade happens or finishes its chain reaction, fibrin is formed, and that fibrin is essential to clot formation. More fibrin means more clots. So more coagulation cascade, or a more efficient coagulation cascade, or a faster coagulation cascade, means more fibrin, which means more clotting. Now, as you can imagine, more clotting can be really important if a patient is bleeding. That's how we provide hemostasis and essentially stop the bleeding. But as you can also appreciate, clotting isn't always a good thing if we have too much of it. Remember, clots cause strokes, myocardial infarctions, DVTs, organ damage, and so on. So that's why it's so important that our bodies have an optimally operating coagulation cascade that kicks in and fires up when we need it a lot and we need a clot. Maybe we have tissue or vessel injury, but we also want a coagulation cascade that knows how to slow itself down or stop when we no longer need any more clots. So considering all of that, this is actually a very abbreviated representation here of what actually happens in the coagulation cascade. I know that's probably hard to believe that this is the simple version, this image here, but what is not shown here are additional factors as well as some of those helpful feedback loops that keep the coagulation cascade kind of in check so that things don't get out of hand. And some of those controlling mechanisms are things like antithrombin, where as the cascade is going, mechanisms are set up to slow it down. And that sounds kind of odd, but you now hopefully understand why that would actually be important in something as a delicate balance as bleeding and clotting. Okay, so we understand that the coagulation cascade results in fibrin, which results in more clotting, but how does this cascade work? And I will mention that this is a very intimidating diagram, and it's really only here to convey to you some key points. But this isn't the kind of information that you would be tested on in a nursing exam or on the NCLEX. So let's just ask in general, what's happening? Well, taking a look here, the story begins with this concept of factors. What are factors? Well, these are little proteins actually floating around in your bloodstream. Remember, your bloodstream has red blood cells and white blood cells and plasma and electrolytes and so on. You have factors also floating around. And they're kind of in this resting state all the time. These factors are deactivated or inactive. I think of it as sleeping factors. They're inactive, just floating around in your body, not doing a whole lot. However, these sleeping or inactive factors can be woken up or activated. And an activated factor can then activate another special factor, which activates another special factor. And it goes on, and there goes our chain reaction, all the way to our end result, fibrin. So you can see here, all of these factors, factor 12, factor 11, factor 9, factor 10, factor 2, factor 1, and factor 7. Think of all of these as little proteins sleepily floating around in your bloodstream, not doing a whole lot. And they're not, floating around in a specific order. They're just floating around. But then wait, something happens. Let's say you get a paper cut and we now have vessel damage. It would be really nice for that bleeding to stop, right? And we need some local clotting to go on in your finger. So what happens? That damage actually wakes up or activates our sleeping or inactive factor 12. Now that factor 12 is awake, it becomes factor 12A, activated factor 12. It can actually now activate that sleeping factor 11. and that now activated factor 11, or factor 11A, activates factor 9, who, through a little help from factor 8, activates factor 10, and then factor 10, now awake and activated, activates factor 2, and now factor 2 activates factor 1, and as it turns out, activated factor 1 is called fibrin, our end result of the coagulation cascade. Now, what I've just described is the intrinsic pathway to fibrin. But there's another way I can get to fibrin. There's another way I can complete this coagulation cascade, and that's the extrinsic pathway. And as you can see over here, an inactivated, sleepy, not active factor 7 floating around in the blood is woken up or activated and turned into factor 7A by something called tissue factor or trauma. So if a patient has an increased secretion of tissue factor, which happens during trauma as well as other events, which we'll get into those details later, that will turn factor 7 into factor 7A or activated factor 7A. So from there we see factor 7A can also activate factor 10, and then now we follow the same common pathway as described before, where factor 10 activates factor 2, which activates factor 1, and we end in fibrin. Now, one more thing to mention. Notice that a couple of these very important factors in the common pathway have names. Some factors just go by their number. Factor 9, for example, is factor 9. But others get names in addition to their number. So factor 2 is actually also known as prothrombin. And when that sleepy inactivated prothrombin gets activated, it becomes factor 2A, which also gets a name, thrombin. So don't be fooled when you hear prothrombin, it's just another name for factor 2. And when you hear thrombin, it's just another name for factor 2A. And the same goes for fibrinogen. That's just another name for factor 1. And fibrin, this really important result of the coagulation cascade, fibrin is actually just activated factor 1. So the last thing that's really important to understand is this. What happens if we don't have enough factors floating around in the body? Let's say you get that same paper cut and you need some local coagulation cascade to happen so that you have fibrin available for clots. But what if you're really short on factors? If we're essentially missing the links in this chain reaction, or we have a really low supply of links to the chain, then this whole cascade isn't going to happen as quickly, right? We need factors to be readily available in order for this to happen. And if they aren't, then that really slows things down and we can't make as much fibrin. So now that we have a pretty good understanding of some of the basics of the coagulation cascade, let's clarify a few important points that are really important to understand when you start to look at dysfunctions or pathologies with the coagulation cascade. So one tricky thing can be trying to keep it straight in terms of too many factors, not enough factors, which puts me at risk for bleeding, which puts me at risk for clotting, what do the lab values look like, etc. So let's break this down for a second. So if I have a patient who I draw their blood, we send it to the lab, and after analyzing it, they tell us this patient is low on specific factors, like they're low on prothrombin or they're low on factor 8. There's not as many of these factors floating around in their bloodstream. If we have low factors, then the coagulation cascade is going to take longer. It's going to be slower. And if this really important coagulation cascade is taking longer and is slower and takes more time, then we're going to produce less fibrin. And if I have less fibrin, I am less able to form clots. And if I'm less able to form clots, I am at higher risk for bleeding. So bottom line, what this should remind you of is fewer factors means higher risk of bleeding. So let's look at some of the logistics of this process, right? So if we want to know how long it takes to get through this coagulation cascade, there's actually specific lab values that can be ordered that tell us how long it takes. And there's two main lab values that we often look at. These are not the only ones, but these are some very common ones. The PTT or APTT, so partial thromboplastin time or activated partial thromboplastin time, this is a lab value that tells you how long it takes to get through the intrinsic pathway. And it's actually reported in seconds. So you can literally think of it as the time it takes to get through the coagulation cascade. And then the PTINR will tell us about the extrinsic pathway, how long it takes for the coagulation cascade on that side of the coagulation cascade to get to fibrin in a sense. Now, both of these are reported in time. So if I have a high PTT or APTT, or if I have a high PT-INR, all that's telling me is it's taking longer to clot. And if it takes longer to clot, then I'm going to be at a higher risk of bleeding. Now, if the opposite happens, if I have a very low PTT or APTT, or I have a low PT-INR, that means we are taking very little time, very low amount of seconds, right? Very short time to get through this coagulation cascade, meaning I'm very quickly forming a lot of fibrin, so I'm much quicker at clotting, and therefore I'm at higher risk of clotting and all of the problems that come with too much clotting that we talked about before.